Over the past few years, mRNA vaccines have transformed from a theoretical concept into one of the most remarkable medical breakthroughs of our time. This revolutionary technology has not only reshaped our approach to fighting infectious diseases, but also opened entirely new doors for treating cancers and chronic illnesses. In this presentation, we will explore what mRNA vaccines are, how they work, what makes them so powerful, and where this transformative platform might lead us in the near future. In this presentation, we'll explore the fascinating world of mRNA vaccines. We'll start with an introduction and mechanism of action, then highlight their key advantages and development process. Next, we'll look at major applications in infectious diseases and cancer therapy, followed by current challenges and future perspectives. Finally, we'll conclude with how mRNA vaccines are transforming modern medicine. Messenger RNA, or mRNA, is a natural molecule found in all living cells. It serves as a temporary genetic instruction, a code that tells our cells which proteins to make. An mRNA vaccine uses this same principle to safely train the immune system. Instead of introducing a weakened or inactivated virus, it delivers a small piece of synthetic mRNA that encodes a viral antigen, typically a surface protein found on the pathogen. Once injected, our own cells read this genetic message and produce the harmless protein. The immune system recognizes it as foreign, learns to attack it, and builds lasting protection, all without any exposure to the actual virus. The process begins when the mRNA is encapsulated in lipid nanoparticles, tiny fat bubbles that protect the fragile RNA and facilitate its delivery into human cells. After injection, these nanoparticles fuse with cell membranes, releasing the mRNA into the cytoplasm. The cell's ribosomes then translate the mRNA into the encoded viral protein. These newly made proteins are presented on the cell surface, signaling the immune system that a foreign invader has arrived. This triggers both antibody production by B cells and activation of T cells, creating a robust and long-lasting immune defense. Once the job is done, the mRNA naturally degrades, leaving behind no trace except immune memory. The advantages of mRNA vaccines are extraordinary. Traditional vaccine development can take years, sometimes decades. In contrast, mRNA vaccines can be designed within weeks once a pathogen's genetic sequence is known. Because no live virus is involved, there is zero risk of infection or reversion to virulence. Moreover, the mRNA platform allows rapid updates to address emerging viral variants, a feature that proved essential during the COVID-19 pandemic. Manufacturing is also highly standardized. The same basic process can be used to produce vaccines for different diseases, simply by changing the genetic code. The development of an mRNA vaccine begins with selecting the target antigen, usually a protein that can elicit a strong immune response. Researchers then design a synthetic mRNA sequence optimized for stability, translation efficiency, and minimal immunogenic side effects. Next, the mRNA is packaged into lipid nanoparticles to ensure protection and effective delivery. In preclinical studies, scientists evaluate how the vaccine performs in cell cultures and animal models, testing both safety and immune activation. Once promising results are obtained, the candidate proceeds through clinical trials. Phase 1 to assess safety, Phase 2 to optimize dosage, and Phase 3 to confirm large-scale efficacy before regulatory approval. The most prominent example of mRNA vaccines in action is, of course, the COVID-19 vaccines developed by Pfizer Biotech and Moderna. Their success demonstrated not only the speed and efficacy of mRNA platforms, but also their scalability to billions of doses globally. Today, research is rapidly expanding toward other infectious diseases such as influenza, respiratory syncytial virus, RSV, Zika, and even HIV. This flexibility means that in future pandemics, we could respond within months, developing tailored vaccines far faster than ever before in human history. Beyond infectious diseases, one of the most exciting frontiers for mRNA vaccines is oncology and cancer therapy. mRNA can be engineered to encode tumor-specific or neoantigens unique to an individual's cancer cells. When delivered into the body, these vaccines teach the immune system to recognize and attack malignant cells with precision, minimizing harm to healthy tissues. Clinical trials are currently exploring mRNA-based vaccines for melanoma, pancreatic cancer, and lung cancer. This personalized approach marks a new era in immunotherapy, where treatment is custom-tailored to each patient's genetic profile. Despite their enormous promise, mRNA vaccines face several challenges. First, mRNA molecules are inherently unstable and can degrade rapidly, requiring ultra-cold storage conditions, a limitation that complicates distribution, especially in low-resource regions. Second, certain formulations may trigger unwanted inflammatory responses, which researchers continue to refine through better lipid compositions and RNA modifications. Lastly, the high cost of production and the need for specialized manufacturing facilities present additional hurdles. Addressing these challenges will be critical to making mRNA vaccines accessible and sustainable worldwide. The future of mRNA vaccines is incredibly promising. Scientists are developing next-generation platforms such as self-amplifying mRNA, which can replicate within cells and achieve higher efficacy at lower doses. Thermostable formulations are being explored to eliminate the need for deep freezing. We may soon see combination mRNA vaccines that protect against multiple diseases in a single shot, as well as therapeutic vaccines for autoimmune disorders, allergies, and metabolic diseases. Equally important is ensuring global equity, making sure that low- and middle-income countries benefit from this technology just as much as wealthier nations. To conclude, mRNA vaccine technology represents one of the most significant achievements in modern biomedical science. It bridges the gap between prevention and therapy, offering a flexible, powerful platform capable of responding to both emerging pathogens and complex diseases like cancer. While challenges remain, the potential of mRNA to reshape human health is undeniable. As innovation continues, we are witnessing the dawn of a new era, one in which genetic information itself becomes the foundation of medicine. Thank you for your attention.